Hey y'all, I thought I would record just kind of an informal recap of what we went through with this PowerPoint on biomolecules this week in class, just in case anyone wanted to use it for some review or if anyone missed class. So just to start off, the picture up here on our slide shows us what some of the main building blocks of the cell are, including sugars, which we will talk about a lot more when we get to metabolism, fatty acids, which we'll be talking about today with our phospholipids and our cell membranes, amino acids, which make up proteins, which do pretty much all the stuff that the cell needs, and nucleotides and nucleic acids, which we will talk about a lot more next semester when we get to genetics. So just to start out with a little chemistry background here, the things that we care about the most from chemistry are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. The other things are very important, but when we're talking about the things in our cells, these are the most plentiful elements. It is important when we're looking at the chemical structures to remember how many bonds these particular elements can make. So if you look at this chart here, the top row is really the most important for what we want to do. So carbon can make 4 bonds, hydrogen makes 1, oxygen makes 2, nitrogen makes 3, phosphorus makes 3, and sulfur makes 2. There are possibilities for expanded bonding with phosphorus and sulfur. We do see this sometimes in, for example, our phosphate groups in our nucleic acids with phosphorus. If you look over here on the side, these are some chemical structures of some of the structures that we just mentioned are important for our cells. One thing I do want to point out is how these structures are drawn. So if we look at the protein structure here, for example, there are 2 lines that come together here to a point, but it doesn't indicate what element it is. So anytime you see these 2 lines come together, that implies that there's a carbon there. And now if we count the bonds on this carbon, it has 1, 2, 3, 4, because it has this double bond with oxygen. And so it is full. So we can assume that that is the appropriate structure. However, if we look down here at the nucleic acid, this carbon here that is bonded to nitrogens on both sides only has 2 bonds that you can see. And so in this shorthand version, we can assume that if it's not drawn, that the carbon is still full and the other missing bonds are to hydrogen. So the shorthand ways where you'll see really not as many hydrogens as are present because they are everywhere really, especially when we have a lot of carbon hydrogen chains and things like that. So often you'll see this kind of shorthanded version of the chemical structure. Okay, so when we talk about lipids, they're a pretty diverse group. They're hydrophobic molecules, which we'll talk about later. They're definitely an important building block for the cell, and they have some characteristics that allow them to come together into the structures that we see. So one of the most important things we talk about with chemical structures for biology are their interactions with water. So this has to do with polarity and electronegativity. So a review here, if you remember from chemistry, talking about electronegativity, you can think of it as basically how strong of a pull the element has on the electrons when it's sharing them with another element. Or the electrons, I think of it like they hang out closer to that one and further away from the other one. Electronegativity, you can see these like little angry faces down here. So fluorine, for example, is a lot more electronegative, so it has a bigger, little angrier face here. If it was in a bond with carbon, for example, there is a pretty big difference in the electronegativities. So in that case, the electrons would be more around the fluorine, which would make that more negative because it's pulling a negative towards itself. And the carbon that it would be bound to would be a little bit positive because you're pulling a negative away from it. So think like in math, you have a minus ends up being a positive. Carbon and hydrogen, however, have pretty close electronegativities, and so we consider them a nonpolar bond. So that means that the electrons are shared fairly equally between them. Same thing if you see a carbon-carbon bond, obviously then their electronegativities will be exactly the same, and that would be definitely a non-polar bond. Now, if you think of what one of the most common molecules that are polar that we see in our cells is water. So we also characterize molecules by whether or not they can interact with water. Polar things can interact with other polar things. So any of the molecules we see that are polar, we also consider them hydrophilic, which means water-loving, so they can interact with water. On the other hand, if they're hydrophobic, think water-hating, they don't have any of those partial charges from their electronegativities, and so they would not have any way to interact with the water by their charges. So if we look at fats, for example, they have a great number of carbon hydrogens in these chains, like these fatty acid chains. And you can see how they can come together to build bigger structures here. But these carbon hydrogen chains are very nonpolar because they only have carbon and hydrogen bonds. If we're characterizing a molecule, it's not necessarily a yes or no question of is it nonpolar or hydrophobic, yes or no, but how hydrophobic is it? So if I'm comparing these two, these chains are hydrophobic in both places, but this molecule, because it has a lot more hydrophobic chains attached to it, we would say is probably more hydrophobic. So this is a zoomed in picture of a phospholipid. So these are the structures that make up the cell membranes. You can see the little cartoon version of it here. I always think of it having like little wiggly legs there. And so the head, the top of it is polar. It has a variable group up here. So what you see on the top can be different for different phospholipids, but phospho means phosphate. So they do all have this phosphate group, which is negative, which helps with its polarity. And then they will have these fatty acid chains, which are very nonpolar. So in other words, you have a molecule that is part hydrophilic or water-loving and a molecule that is also part hydrophobic here or water-hating. And so this characteristic will help determine how these phospholipids function in the cell. An example here of this is just a carbon-hydrogen chain. We tend to say sometimes that carbon and hydrogens repel water. What really is happening is that the water molecules can all interact with each other, but because they can't interact with this, there tends to be a space around them. So it's almost like they're pushing them away if you would look at them visually. If you take all of those phospholipids and let's say you drop them in a bunch of water, they will naturally form into some structures like these micelles on the left. And that's because the heads will naturally orient themselves to face the water, which they could really do anywhere, but because the tails are hydrophobic, they'll tend to cluster together because they're being, think of like not interacting with the water, so they each get a space around them. And so if they hang out together, the space basically just turns into one big space. And so you get this kind of bubble or micelle here that's forming with the one layer. Now, if you think of this flattening out and making like a bigger sheet, you see what we see here, which is really more like our lipid bilayer, which would go around the whole cell. And you can see the zoomed in picture here. The hydrophilic heads are on the outside of the cell. If this is a cell, for example, facing the extracellular matrix. So that would be like all the stuff that's outside the cells in your body. And then the inside would be interacting with the cytoplasm, which is basically the stuff in your cell that's holding it all in together. And both of those are very aqueous or filled with water. This should look kind of familiar to you. So if you look at the left here, this looks a little bit like what we saw with the oil droplets. So when we looked at that kind of the origins of life, when we did the TED Talk, very similar things here. So you can see these artificially formed liposomes. And again, this is how we can see a membrane form. And this looks very similar to the cell membrane. So you have water on the inside, water on the outside, and then this double layer where the inside, where all these tails are, is very hydrophobic. So it's all hiding from the water. One of the things that's important is to think of your cell membrane or any membranes that you see as being very dynamic. So dynamic meaning that when you look at these purple phospholipids, they're moving all over the place, but only within their layer. So they can move all around in their layer, so they've moved to different places, but it would be very rare for them to flip to the other layer. So for example, the green ones will never flip into the layer with the purple ones. The only time we really see this happening, this flipping with the cells, is when they're going through apoptosis. which is programmed cell death. So it's kind of an interesting thing that your cells will program a way to disassemble the membrane when the cell is being killed, and that's basically to flip the membrane inside out. So you can see that that would be very disruptive. So part of why we talk about the membranes is because it is a barrier that is selective for the cells or the organelles or wherever you see them. And so it will determine what solutes or molecules can go through the membrane and what can't. And that's largely due to the types of phospholipids that are in the membrane, and also any of the other proteins or molecules that are in the membrane along with the phospholipids. So the three major things that will determine this are fatty acid saturation, which we'll talk about in a minute, the length of the fatty acids, and the cholesterol content. So this is a really good kind of list of things that can go through the membrane. So if we look at the permeability here, so on the right, on our phospholipid bilayer, things like oxygen, carbon dioxide, and nitrogen can go through the membrane. Water, indole, and glycerol can also go to some extent through the membrane, but things like glucose, sucrose, and ions cannot. And this should make sense. So glucose and sucrose, they might have a lot of carbon hydrogens in them, and so they might be able to interact with some of the hydrophobic parts, but they may not be able to interact with the hydrophilic parts where you would see some polarity. The opposite's true for the ions. So the ions would likely interact with the heads because there's some partial charges in the heads because of the electronegativity, but because they're charged, they couldn't interact with the tails. So the fact that this bilayer has these two different characteristics is what determines that it's selectively permeable, because some things cannot go through both layers of the phospholipid bilayer. Okay, so to talk about saturated and unsaturated, this is usually something people have heard from a nutritional standpoint. In this case, we're not looking at is it good or is it bad, but just what does it do for our membrane? So if you remember talking about that shorthand of how we have carbon-hydrogen chains, one of the things that you see sometimes is a double bond between carbons. And so you can see that where there's all these carbon hydrogens, there's like a zigzag shape, but where there's a double bond, it basically puts a kink or a turn in the structure. So these carbons could have bound with hydrogen, but they didn't. They're bound with each other instead. So because they're not saturated with hydrogens, we consider them unsaturated. If they had hydrogens on them, it would look more like the one on the right, so more of this straight shape, and it would be saturated. The reason that we care about this in terms of membranes is because if you have a lipid bilayer with no unsaturated fatty acids, what you get is sort of, if you think about this like a group of pencils, if you tried to thread something through this cluster, it would be kind of hard. There's not a lot of space between all of these pencils. On the other hand, if there's a lot of unsaturated fatty acids where there's all these turns and things that they look kind of like a tangle, if you look at this, it looks like a mess, but there's a lot of open spaces in the hangers or between the hangers that you could actually weave something through the matrix. And so the important thing is to have a balance of these two so that you have the appropriate amount of permeability for your membrane. Just give you some context of saturated and unsaturated that might make sense to your other parts of your life. So butter is very saturated versus safflower oil, which is more unsaturated. And they do tend to say that the unsaturated is a little bit better for you health-wise. But again, in terms of cells, it's more about the balance that you would want to strike. The other things that we see sometimes in the membrane are steroids and cholesterol. So you don't really need to know too much about the chemical structure of these, but if they are in, so for example here, the steroids, if they're going to be in the membrane, they likely have similar makeup to the phospholipids. So the steroid rings, for example, they're going to have a polar aspect, and then they'll have a nonpolar attachment to them, and that will allow them to basically fit in the membrane along with the polar and nonpolar parts of the phospholipids. You also see the cholesterol, it looks kind of like this. So you can see that it makes some space. It affects, again, basically the permeability of the membrane and also how fluid it is. So if you have a lot of cholesterol, it may be a little bit thicker of a membrane or more stable, or if we're looking at basically how fluid it is, how wiggly it is, it matters that you have the right amount of cholesterol. If you have too much or too little, your membrane could be either too rigid or too kind of wiggly so that the cells don't maintain their appropriate shape. Okay, so the big thing for what we're talking about, especially related to metabolism, is that membranes are helping with compartmentalization. So this is true both in terms of the larger cell, but also in the membrane-bound organelles that we see in eukaryotes, such as the nucleus, the mitochondria, the chloroplast in plants, and those are really important for metabolism. The molecules being concentrated together, so like the proteins that can act as enzymes and receptors, you can see them in this membrane. Having them close together in the membrane is important, and also having more membrane gives you more places to have these proteins. So for example, if you think of a picture of a mitochondria, the folds inside give a lot more surface area because you can fit more membrane in that organelle. And that means that more proteins that do metabolism can fit onto the membrane. And that helps us be more efficient in our cells. To give a little context of that, so here again is the mitochondria. You can see all the folds in it. So everywhere there's membrane and this inner membrane, there are enzymes that are helping do metabolism. The same thing is true in chloroplasts. The stacks called thylakoids here, I think of them a little bit like a pita made of membrane. So again, a large increase in surface area that's usable for metabolism, just in this case, photosynthesis. We also see the nucleus that has a membrane, the endoplasmic reticulum, and the Golgi bodies. We will talk about more later when we get to the regulation of proteins because they help with protein transport and packaging. And then one of the final things to look at is the bacteria. So bacteria have this outer and inner membrane, which should help us remember the endosymbiotic theory, because we thought that basically if a eukaryote or a primitive prokaryote basically ate a bacteria cell, that's how we got the organelles that had these double membranes inside, so like the mitochondria. So over time, they might have started as like an aerobic bacteria, but they became part of the cell with a slightly different function. So these membranes are also very important. Hopefully that helped clarify some things for you for review. And as always, let us know if you have any further questions.